In this video we will discuss about the role of this C1GalT1 in the PDAC progression. So as we have discussed in many of our previous videos that the PDAC there has been a aberrant glycosylation that is observed. So the hypothesis of this paper is that the deletion of this C1GalT1, which is an enzyme that helps in the O-glycosylation, will leads to the faster PDAC progression. As in the PDAC, that it has been observed that there is a lot of aberrant uh, aberrant glycosylation observed in terms of truncation of this O-glycan on the MUC16 protein. So they hypothesis that it could be because of this C1GalT1 disruption that might have occurred in the pancreatic cells that will lead to the this PDAC metastasis or PDAC higher progression. So they have used the mice, PDAC mice that has this KP crass knockout and TP53 knockout. Along with that, they use the similar mice model, but they have also knocked out this C1GalT1, and to this they call that mice as KPCC mice. And they can find out that only in the 10 weeks the KPCC mice has higher tumor incidence compared to the KPC mice at 28 weeks. Thereafter, they also check this histology analysis, and they have find out that there is a faster rate of PDAC progression in the case of this KPCC mice compared to this KPC mice. As we can see here, only in 10 week there is a uh, tumor progression can be seen, whereas in the KPC mice there is not much tumors has been observed. And thereafter they also check whether it will impact the survival rate of the mice or not, and they find out that the median survival rate of the mice got reduced in case of this KPCC knockout compared to the normal KPC knockout. And this study are being done on 11 based on 11 mice models. And later on they also stain or histology analysis of the pancreas, and we can see that only in 10 weeks the tumor progression in the pancreas is very much higher compared to the case of KPC at KPC histology analysis of the pancreas, KPC mice histology analysis of the pancreas at 30 weeks. That can be seen here. And let us understand what is this C1GalT1 and what is its normal role. So this O-glycans usually occur in serine and theobromine site, and after that the C1GalT1 helps in the addition of this galactose and or helps in the addition of this T antigen. We can say that this is the T antigen, and thereafter the sialic acid with the help of ST3-Gal1, the silyl or sialic acid is added to this structure, and this whole structure is known as silyl T antigen. And when this C1GalT1 is not there, then this TN antigen is directly silated through this ST3-Gal NAC1, and this is known as silyl TN antigen. So in case of this pancreatic cancer, what has been observed is that there is increase in the amount of this TN antigen are been found. So because of that, the hypothesis that it could be one of the reason why the increase in the amount of silyl TN antigen in the case of pancreatic cancer is because the C1GalT1 might not be functioning properly. So that's why they decided to check whether this C1GalT1 has any role in the PDAC progression or not. And this is just one figure of the whole research, which shows that the PDAC, the C1GalT1 knockout helps C1GalT1 knockout helps the PDAC progression or supports the PDAC progression. So in the case of the normal knockouts that are been observed when in the case of pancreatic cancer, normal knockout mice will show a pancreatic tumors in 28 weeks, whereas the KPCC mice, that is all the normal knockouts in the pancreatic cancer plus the C1GalT1 knockouts, shows the pancreatic tumors incidence much faster, that is within 10 weeks. So this figure indicates that this C1GalT1 leads deletion of C1GalT1 or knockout of C1GalT1 or disruption of C1GalT1 leads to this faster PDAC progression. So I hope you have understood how the role of C1GalT1 in this PDAC progression to some extent. If you need to understand more about this C1GalT1 role in the PDAC or pancreatic adenocarcinoma, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.